It's a real pleasure, Genese, to have this paper for discussion today in the reading group. It's the second time I've had the opportunity to engage with it, and I really love the way that you're taking seriously kind of the subjectivity and phenomenology of the flea alongside the bacteria, Yersinia pestis, alongside the different rats. You're looking at multiple species and also practices of multi-species recognition. And I really just wanted to start by zeroing in on one of the key concepts you introduce with the paper, which is virulence as an epistemic thing, something that's somewhat fuzzy. So maybe if you could just say a little bit more about virulence and how it comes to being in these multi-species worlds that humans are part of, but kind of in some ways tangential to these other, commensal and symbiotic and parasitic relationships that you describe.
Thank you, Evan. Thank you, Rachel. It's a pleasure to be here with such an international learned group. So I do look forward to people's comments and input as this takes shape. The concept of virulence, thinking about it as an epistemic thing, much as you did, Eben, in your discussion of species in a praxeographic study of species, how we can take, Reinberger, in speaking about epistemic things, is looking at actual material objects and that are sort of fuzzy and unknown and open to, investigation and interpretation from multiple sort of disciplines. And he writes that they, in studying or getting to know that and trying to anchor these into some sort of reference, they sort of enable sort of a river of discovery, or just a really fruitful way of thinking about something. And so just some background, as Evan mentioned, I presented this paper at the Anthropological Association meetings, and our panel was about taxonomies. So taxonomies of the present, it was called. And I was interested in Evan's work and multi-species, the ethnographer's work in thinking about trying to move our perspectives and our lenses into other beings, and thinking about taxonomies of non-human lives, how they make choices and preferences. So I was thinking, when I get back to virulence, because I sort of came to this path of virulence through another door, I was really interested in the fact that fleas have their preferred mammal hosts, and they have, preferred blood meals. And in my research on the plague in Madagascar, this is very important because there's a species of flea, Xenopsilla chiapus, that is the main vector of the plague. And it's a rat flea, but it also will feed on humans. And then we have the zoonotic spillover event. So I was speaking with an entomologist there and thinking about the choices of fleas, their preferred hosts, their preferred mammals, and what happens when they take their second choice, a human, take a bite out of a human because their first choice, their host, dies. So that's kind of where I was thinking about it, and then and reading up more about fleas and their blood meals and the sort of their parasitic relationships to their hosts. Sort of this idea of virulence from a different standpoint emerged, and that was what happens inside the flea when it feeds on the blood of a rat. And sort of this really interesting relationship emerged, that the physiology of the flea, the anatomy of the flea, made it a very efficient vector of the plague because of the way it's built, because of what happens in this relationship with the rat. And it just became apparent to me that those things really mattered, and the way scientists were talking about virulence in other contexts were not really addressing those sort of anatomical and kind of relationships, but it did play into how much plague bacteria was injected into a human being and why. So, and then Reinberger's work just kind of was useful to me to think about, depending on what your entry point is or where you start thinking about a subject like this. And I'm starting from the body of the flea versus, the pathogen itself. Or I know there's a lot of talk now with COVID about these different variants and how virulent they are, and endemicity and all these things, sort of in debate about, does endemicity mean less virulence? And there seems to be confusion about that. And so this was in the back of my mind as well, in coming about this. So yes, so I thought virulence seems to be tied in this case to how much blood is sort of penetrates into human flesh as far as virulence to humans go, but it's also the bacteria is very virulent to fleas, and they die and they suffer, and they when you read about it, you kind of imagine their experience of ingesting this bacteria. So that was that's what inspired me with Reinberger, and with your work too, Evan. I'm thinking about how these ideas take shape and what is their materiality.
Thanks, Genese. And if anyone else has a question, like usual, feel free to raise your hand, just unmute yourself, or put a question in the chat. If nobody else has another question, Warwick, go for it.
Thanks, Evan. I always have a question, as you know, but anyhow, a number of questions usually. Thanks very much for the paper and the little discussion then. I liked it very much. It reminded me a little bit about what I think was Celia Lowe said about bird flu, about how if you think bird flu, influenza is bad in humans, you should see what it looks like in chickens. I mean, for fowls, it's actually terrible. It's absolutely appalling. And we tend to think of it in terms of a threat to humans rather than a threat to other creatures. And so, but the other thing that it reminded me of was the studies of Frank Fenner and Francis Radcliffe of myxomatosis in Australia in the 1950s, when they looked at the infectivity of various mosquitoes, trying to work out how, well, the avidity, in a sense, of various mosquitoes for the myxoma virus, and how their mosquito physiology was very important in transmission. And that was an enormous study that they did of myxomatosis, which is basically a rabbit disease. And in fact, I think Robert May said it is still the best modelling of an epidemic that's ever been done. And so that might be of interest too.
That would be really useful, yeah.
Yeah, but more to the point, in a way, I've been reading recently a lot of the work of W.W.C. Topley from the 1920s, very important British epidemiologist, as you know, the fellow who coined the term herd immunity 100 years ago. But he has this argument, which I don't understand at all, about whether we should be calling it virulence or infectivity or pathogenicity. And he said they're each quite different. I don't understand it. But I mean, you've chosen the term virulence, but I was wondering whether you see that as distinct from pathogenicity, infectivity. How do they relate? Are they the same? Is this just a word game? Perhaps it doesn't really matter. But I'm puzzled by the use of these different terms.
Yes, I think that there's a lot of debate about definitions, and it's very discipline dependent as well. So I think that pathogenicity is often enfolded within the concept of virulence for many people. Virulence often is, again, depending on what discipline you're you're speaking from, it's often tied to the idea of damage done to a host or damage done. The infectivity doesn't necessarily mean damage, it could. For medical scientists, virulence really has to do with mortality rates and deaths, but not necessarily for microbiologists, say. And the more that you look into this literature, these theories of virulence transmission trade-off and all the debates within that, and all the different studies that have yielded different results and sort of found that this model doesn't work when you're looking at these species. And of course, the classic model developed in the 1990s on virulence transmission trade-off seemed to not apply at all to the insect world and relationships to insects, and same with plants and relationships to viruses, for example. So I think you're right to say that there's a lot of fluidity in those terms, and really, it has to be well defined, whoever's using the term virulence, and to what purpose. It's very much used in discussions of the plague, and it's an interesting contrast to discussions of COVID and endemicity, and this idea of if it's endemic, it's automatically means it's becoming milder, which isn't the case. With the plague, for example, is endemic and very virulent and isn't changing. It's just sort of settled into the environment now. And I think that many scientists thought about virulence in terms of something that evolves. It goes through a process of evolution. And that's an interesting way to think about it, maybe to distinguish it from these other terms. But I appreciate your references too. I want to check out that Radcliffe and Fenner work on mosquitoes, because that's really relevant too.
I wrote an article for a journal on that. I can't remember the name of the journal now, but anyhow, about rabbits. It was a couple of years ago. Studies in History and Philosophy of Science. No, it wasn't that. It was, I don't know, History and Philosophy of the Life Sciences or something, I don't know. One of those multiple journals. But actually, as you were speaking, I just thought as a follow-up that you made a nice distinction there in terms of endemicity and epidemic, between virulence and infectivity, perhaps. And that there may be a confusion now that basically changes in infectivity mean changes in virulence, which they, as you say, don't necessarily. And so that could be actually a useful distinction, as you point out. So thanks for that.
Paco, go ahead.
Yes.
Or go ahead, Genese.
Yeah, it's something I was focusing. I didn't really delve too much into the whole theory of theories of virulence in science, the whole virulence transmission trade-off, because it's complex and it's vast, and I don't know the extent to which I want to get to delve in too much, but I think if I can, just pursue those studies that are about the plague or comparable studies with other insects, I think that would be interesting to see what they cast off, what they retain. And even within one disease, like the plague, where you have these different relationships to the pathogen depending on the species, it seems like even within those relationships, different theories of virulence would apply, like the rat Yersinia pestis relationship, the flea Yersinia pestis relationship, the human Yersinia pestis relationship. So yeah, I have to keep exploring.
Thanks. Paco, go ahead.
Thank you, Eben. Yeah, this is a fascinating paper and in general line of research. Many thanks, Genese. So my question has to do with some of the things that you were mentioning at the introductory comments related to epistemic things and epistemic objects in Reinberger's definition. So I want to talk about the relationship between that and which specialists have been involved in discovering these particularities, these peculiarities about the fleas. If it's mostly parasitologists or entomologists, because I think that there might be a difference in the approach that these subspecialties of biological or biomedical studies allow people to see things. No, so I think that this issue of the behavioral repertoires of these animals is extremely interesting. Not to perhaps even to strengthen some aspects of your argument. So the actual question is, if you have looked into this literature to see who is actually behind, or which subdiscipline is behind these discoveries, and if there's actually much more about these behavioral peculiarities that is known about fleas and phylogenetic or species in the phylogenetic vicinity of fleas. And another loose comment has to do with what I think it's something that is probably something that everybody who reads a paper like this might think about, which is a connection with von Uexküll's concepts about environment, right? When it comes to the fleas. And so I'd like to know also if you have thought about a connection with all this literature that is appearing recently about Jacob von Uexküll and environments for animals. Thank you.
Sorry, you broke up a little bit for me at the end there, so I don't think I heard fully the last comment. I'm sorry, I'm having some connection.
It was about Jacob von Uexküll's views on environments for animals and their perceptions, Umwelts, to use the German word. Yeah.
Yes, I to tell you the truth, so this is a new, like I told Evan at this conference, I don't know how I ended up in the foregut of a flea. I'm an anthropologist, but you never know where this is going to take you. But I have been looking at rat environments and burrow cities and looking at some multi-species abodes of the plague, not only houses where people live in their tombs, but these burrow systems that undergird human settlements. And those too are multi-species spaces where sometimes other rodents, other burrowing rodents will come in and occupy them, or abandoned places, or there's different insect species. And there's it's just a fascinating world there. And it's really relevant to how the plague settles into the environment, literally into the land, but also theories, hypotheses about how the plague has been transmitted or how it survives and persists underground, ideas about the bacteria being absorbed by insects, which are then consumed by rats and other animals in this world, this ecology. So I've been looking at those kinds of Umwelts, more about rats, and this is a newer sort of deep dive into the flea's digestive system, but also I feel like for us, the invisible worlds of insects and what they inhabit aren't sort of readily apparent when you're doing field work, when you're in places. So it's really a reliance on scientists. And that's a great question about who's been publishing this. There's been a lot of biologists publishing what I've read about the fleas, but I'm sure others as well. I have to look at their backgrounds, the authors, more closely. I've done interviews with an entomologist in Madagascar, since that's my field site, and sort of, her knowledge and my interest in what the scientists are looking at and where they're looking at it. So I've been guided by humans to a great extent. And, I want to keep, my mind open to what else I could look at. I mean, actually, Warwick's work on these animated communities and early 20th century scientists looking at zoonotic communities in Africa and elsewhere, looking at these communities of non-human species and their interactions. And I've been approaching my work in that way. So yeah, there's a lot for me to learn about fleas and where they, where they live or where they're hatched or where their eggs open up and these kinds of things. Yeah, there's still, I can't say I'm an expert yet.
Many thanks.
Thank you.
Jiahui, go ahead.
Hi, yeah, thank you for a really fascinating provocation in that piece. So I work in Tanzania with people who work with rodents. And so one question that also comes up is plague. And based on my research and conversations in Tanzania, there's sort of two ways, and I think these two ways come up in your paper as well, in approaching plague and the disease flea, the bacteria flea human relationship. And so the first way is a sort of bacteriological sanitary synthesis, so to speak, where you have the medical as well as the biological aspects of the relationship taking a forefront in explaining how the disease is transmitted. And now in the second way, it's a more ecological approach to the disease, which you've also mentioned just now and in the paper, where the question is less about transmission and pathogenicity, but more about the configuration of spaces between humans and rodents and the fleas. And then the question of the sylvatic rodent comes up a lot in these approaches. The ecological approach is like if the rodents and the fleas are usually found in the wild or in the bush, through what vectors and relationships that they then find their way into the human home and then infect the human. And what was really interesting, and this is the question that struck me while reading your paper, is that I think your paper in untangling the different things that make up virulence, the scales that virulence is operating at is at the molecular level of the hemoglobin, for example, not having the same molecular structure, so that prevents a certain interspecies relationship, all the way up to the scale of the configuration of large spaces, households and burrows and homesteads and farms and the bush, right? So I'm wondering how that scalar leaps that happens in both the scientists you talk to, but also in your own analytical work, if virulence tries to capture that kind of scalar leaps, and that's why it's such an epistemic category.
I love that idea from hemoglobin to homestead. I've been thinking a lot about homestead, which captures both the house and the fields around which people work. And of course, there's a close relationship to seasons of rice cultivation and movements of rats and fleas into the home. I'm sure Tanzania is similar. And I think that's an interesting way to think about the flows of the idea of this thing called virulence. And as far as the homestead goes, I think, I've been thinking a lot about, or sort of investigating more about those sort of moments of contact between people and rats and fleas, the time of year, those conditions. But I haven't been thinking about it in terms of virulence, but simply, those risky encounters as we often think about them. And except when it comes to, I should back up. I'm thinking a lot about architectures of commensal life. That's the sort of the book project I'm working on. And architecture is not only meaning the homes and the tombs that people build, and the commensal life of humans and rats and fleas, but architecture is also in terms of the structures other species build, and even the body as a structure, and how that contributes to risk. So there's multiple scales at work there, but I haven't really been thinking about how to weave or the thread between the molecular level and the homestead, or more than the homesteads, even up to the level of urban planning during the colonial era, where the French colonial state revamped cities because of plague risk. Masonry foundations and walls became very important. So there was a whole, architecture around the plague as keeping it out. So I didn't even know if I would use the anatomy blood thing in this project or just keep it apart. But that's an interesting way to think about it in terms of scale. So thank you. I'm just thinking out loud if I ramble, pardon me.
That's what it's all about in this group, productive rambling. Jack,
productive rambling.
Thanks, really interesting paper. I have two questions on the phenomenology angle. The first is what you sense the beliefs or ideas regarding the various scientists and other individuals you talk to about the life worlds of the fleas, what their senses of the phenomenology of the flea. So one of the figures I've written a little about, who was the first director of the League of Nations Health Bureau in Singapore in the 1920s, had these quite strange asides in some of his books where he talks about the capabilities of fleas in listening to sounds, and even being capable of kissing. And so I wonder what extent, there are those sorts of equivalent perspectives on the life worlds of fleas. And the second is that I came to this article having already read your, I think 2019 work on semiotics of plague. And I wonder if this is also a semiotic issue in a way. The flea in a way reading the signs of sickness on the rat or the human. And if that points to a sort of more than human or multi-species semiotics.
Yes, that's wonderful. That's really interesting. I want to explore this this director in Singapore. What was his name again?
Gilbert Brooke. I've written a little bit about him.
Okay.
He developed the Singapore Bureau of Epidemiological Intelligence and was mostly focused on really practical questions of rat destruction. But in these asides, he talks about, these fleas rubbing their bodies together and this being the equivalent of humans kissing. And it only appears a few times, but obviously an interest of his in a way. Quite interesting.
Yes, very much so. I haven't really looked into it, so there's a whole world of interesting things to pursue here. So in a paper I recently gave Christos Linteras, it was part of the St. Andrews group. I was kind of exploring, and I'm still exploring, proverbs in Madagascar. There's hundreds and hundreds of proverbs, many of them concerning non-human species, and there's quite a collection of rat proverbs and flea proverbs. And so there's sort of ways in which humans not only think about these non-human species to understand their social worlds, but also they suggest that people's close observations of the sort of ecology of the household and where these species are moving and living and interacting with one another, and sort of sometimes it's reflected in the proverbs themselves. So now I'm going to think more about the semiotics of fleas and maybe where humans have seen them and how they talk about them might might give clues into other signs that humans have observed. And the other things in that same article on zoonotic semiotics, I'm interested in how people classify fleas that they live amongst and sort of have to deal with every day. And there's a passage, for example, an elderly couple, they were sort of really afflicted by fleas after a flood had occurred. But they were told by public health agents that the fleas are vectors of the plague, but they somehow understood that knowledge to mean that it was the jiggers that get under people's toes and lay eggs. That was the vector of the plague. And something about the physical egg made much sense to them, that would be the thing that transmits the plague. But of course, that's nothing to do with the transmission of plague. So that's I've been exploring in terms of people's interactions with fleas, and I haven't really delved too much yet into the flea world apart from this paper. But early, late 19th century, early 20th century works about studies of plague transmission, as Simon and others were thinking about how the plague was transmitted. And I do think that there was really fascinating studies in this period about rats, which I sort of their behaviors and kind of a vicarious understanding of what they were suffering, because it was very similar to human symptoms of the plague. But yeah, I really look forward to learning more about possible flea emotions and sensibilities, and what they see or smell, or they must smell, right? Blood, and no, I can identify what blood is appealing to them. But yeah.
Rachel, go ahead.
Thanks, Genese. This is great. I was looking through my notes in your paper, and I think you used the word preference most predominantly, but I was really struck when I got to the end of your paper thinking, just as somebody who writes about food, thinking about taste. And yeah. I'm curious if, there's an element of that comes out in the human subjects blocking of what can be studied in some insects, right? That you mentioned in the paper, but I think it would be interesting. Just going along with what you just kind of the tail end of what you just responded to, maybe open up this avenue of taste, because you use the word preference. But, in the food studies world, taste and taste making often acquired, is something that is acquired. It's made, it's about marketing, it's about consumers. It's a very different framing, and rarely a multi-species one, if ever. So there's kind of an interesting, just fascinating unknown there. And I'm just curious about what it might, if it could open up, or if it does already, is it already something that's being pursued in the lab with the studies that can continue?
Yeah, those are great. The only clue to that is that passage about, for the entomologist in Madagascar, Dr. Murray, the difficulty in studying plague in the human flea, Pulex irritans, because it's finicky, it will only feed on human flesh, and therefore they can't feed it. They don't live long enough in the lab it refuses to eat the baby mice that they give in the lab. So it just, that in itself was sort of led me down this path. It's very interesting to me that it refuses anything else, even would rather die than eat from a mouse, So yeah, what is that? And she may know more about it. I hadn't really asked her the taste questions, or how much, what she knows about that. Just like it could be the properties in the blood of rats and guinea pigs that have that hemoglobin, that property that makes it, makes it appealing. But yeah, this idea of taste making, and that's an intriguing possibility. This is because of the commensal life of humans and rats and tree shrews and mice, and the proximity of fleas to humans in those encounters. Maybe there was an acquired taste to feed on humans when all else fails. So yeah, it's a really interesting way to think about it.
I've got a question on that point, and I see Warwick also has a hand up. So I would like to try to return to this idea. It's related to taste of recognition, differentiation, and classification in these multi-species worlds from the embodied perspective of the tick, and building a little bit on Paco's question about the Umwelt and what Jiahui was saying in relation to hemoglobin. I guess I'm wondering what kinds of beings these fleas are recognizing, and also if the Pulex irritans, is that right? The human, the one that likes people, is it just because of the lab apparatus that it's thought to only like people? Like, what if you offered it, say, like a baboon or a rhesus macaque or an orangutan? Like, is that is that kind of configuration going to open up the category of being which is tasty, or does it have something to do more with like the cues that von Uxkall was interested in? So like, he's looking at butyric acid, warm hairy membrane. Maybe is it a hairless membrane that this particular flea is interested in? If you shave these animals, so maybe what are the conditions of possibility for producing that bite? And yeah, maybe it's also a nested question about like political economy of science in Madagascar, and has anyone tried lemurs, for example, or
shaved lemurs?
Yeah.
Yeah.
For lemurs.
Those are great questions. I should say that
Go ahead.
Sorry, I lost your final words again too. I'm just kidding. Yeah, again, so the Pulex irritans, they tried to feed, just in terms of hair, those little baby mice are pink bald little things, It's also interesting in the lab, in the insectarium, they did not allow me to take any photos. And I thought that was really interesting. They did not want me taking photos of the little baby squirmy mice as food, like as though it was, a moral problem for me to record that. So that was fascinating to me. So I could only describe it with words. Yeah, I think I don't know how many other species the entomologists tried to feed the Pulex irritans to. That is really fascinating to think what other, of all the different scents and components of other species' blood and skin and hair, whether some other appetite might be triggered. I don't know. Yeah, it's a really fascinating question. I also don't know what species feed on lemurs, to tell you the truth. I don't know if they have their own unique species or multiple kinds. So that's another question, taking me into lemur land.
Thanks, Genese. Warwick, go ahead.
Right, yes. It's all starting to sound rather anthropomorphic though, isn't it, really? Fleas taste, they eat, they like things, they may even kiss. So I just wonder, and Evan may have a comment here too, whether this is what distinguishes multi-species studies from ecology. in the sense that in multi-species, a lot of multi-species studies, there seems to be a sort of almost reflex anthropomorphism going on quite often. Thinking of some of my colleagues working on pigs and so on, where pigs start to be like baby humans in a way. And so is that unfair though? Maybe it is.
I think that, yeah, ecologists do anthropomorphize all the time. And that, I mean, that's part of, so reading Charles Elton's work on, well, it was this tropical disease scientist in Kenya in the first half of the 20th century, who was sort of a fan of Charles Elton's work and talking about zoonotic communities. And there's, you know, he talked about, it's like calling the badger the vicar, like they all have their role in the community, you know, and thinking about community really as a human community with, you know, there's the vicar, there's the teacher, there's, you know, and those kinds of metaphors were rich to me because, you know, I'm comparing that to the proverbs in Madagascar and the way people anthropomorphize animals there, these very defined roles and the sort of moral universe of the proverbs. It overlaps in interesting ways with the idea of zoonotic communities. So I think, yeah, I think multi-species ethnography is pretty open and generous about who they read and what and how they consider it. I don't really think there's one way, but certainly ecology gives us a lot of interesting material. And I think, yeah, it does seem to be in terms of our own moral universe. I think the anthropomorphism helps to create those empathies, you know, or imagining oneself in a different world, in a non-human world. Yeah, with the goal of creating those kinds of connections. Absolutely, absolutely.
And since work invited me to say something, maybe I'll jump in. And
yes,
I think, you know, there's a broad tendency in some corners of biology of mechanomorphism, to assume that the machine is like the animal. And, you know, Canguilhem and others have written about that long legacy, going back to Descartes and others, of, you know, comparing organisms to machines of the day, like the clock or, you know, the steam engine. You know, the aim of multi-species ethnography or multi-species studies hasn't been really to impose an anthropomorphic sense on the world, although there is you know, a recognition that anthropomorphism is in some ways an opportunity to empathize, an opportunity to engage with the subjective experience of another or the phenomenology of another, but really it's about being specific. And, you know, that's what I like about this paper. You know, there's a specificity of like the biofilm that causes the regurgitation and the frantic feeding behavior of the flea. And it's also, you know, assuming that all fleas are not flea. You know, it's not like one category of, you know, broad organism, but what you're doing, Genese, is really attentively thinking about how a particular kind of flea is engaged in this practice of recognition, differentiation, in a sense, speciation, and, you know, the phenomenologies of each of these organisms is specific and embodied. So I think, yeah, often anthropomorphism, you know, assuming that others have a human form or human subjectivity, can get in the way. And I think that's in many ways why early multi-species ethnographers were partnering with biologists to maybe get beyond you know, the naive realism that we bring to an encounter with another animal or with a plant or with a fungi. But yeah, at the same time, I think, you know, we find in these critical friendships with scientists, yeah, sometimes it's important to reclaim other situated modes of knowing and apprehending. And so in some cases, it's important to ignore the science. So yeah, I'm really interested in where this paper's headed, and I'd also love to hear more about commensality. I looked it up in OED, Oxford English Dictionary, when I encountered it in your paper, and I didn't realize it was so close to the word companion, in the same way that Haraway is using companion as, messmates, those who eat together at the same table. So maybe could you say a little bit more about commensality in the broader project that this paper is part of?
Yes, well, so again, I'm interested in the idea of dwelling with plague. And so dwelling with plague means dwelling in these multi-species abodes. And this is sort of the foundation of commensal life, especially in this case, with the species that transmit plague, since that's the focus. So unlike companionship, I think companion relationships suggest a kind of equality that commensalism implies a parasitic relationship. So one species is benefiting, maybe not in a harmful way to another, but certainly is benefiting, and the other doesn't have any benefit. And I think that it's most clearly apparent with humans and their commensal rats and the commensal fleas that are on the rats. So there's absolutely no benefit to humans in having rats penetrate their homesteads, but the rats do quite well. So that's my main focus. In different parts of the island, there's the shrew, which is very similar. It looks like a rat with slightly different nose, but they're also can be host to the plague. And I've been interested in looking at kind of like a history of rats colonizing the island and moving about the island always in tandem with humans. I mean, the plague colonized the interior of the island via the roads and then the railway. Once the railroad was built, it really established the plague in the interior of the island. But thinking about rats as ratus, I should say, an invasive species, but they're colonizers, just like humans were, and they have this interesting history and these competitive relationships, just like the Europeans did there. So there was the Norwegian rat, which prefers watery environments, and ratus, which is called a ship rat, is an arboreal rat, although it always has, this is a weird thing that people always say with deforestation. Deforestation has increased rat populations in human settlements, but from all the accounts I've read, rats have always been in human settlements. It's not like they waited for the trees to be cut down before they moved into the village. But when the sewer systems were built and the brown rats moved in the tunnels for sewers and in the underground watery environments, it removed them from close proximity to humans. So then ratus became the dominant host of the plague. So those sort of interesting histories of the rats and the rat species with one another within a context of changing human built environments, I think is really fascinating. There's the whole life history of the rats generations. I find really fascinating to think about it in terms of built environments and what they build. So that's kind of my approach is looking at humans and rats in built environments, not only the environments that humans have built, but also how humans have built around rat built environments, their tunnels, the burrow systems. And I should say that the central problem of the plague in Madagascar in terms of the social history of Madagascar has been around mortuary policies established during the colonial period, and this really interesting hypothesis by scientists that plague bacteria could possibly stay active underground for an unknown amount of time, and therefore people were not allowed to inter their dead, the plague dead, in the family tombs anymore, but they had to be buried in separate graves because there was belief that if people go into the tomb, they could get reinfected with live bacteria. So this rule has stayed in place where people may not transfer the remains of plague victims for a period of 7 years to ensure that the plague bacteria are no longer active. And that has never been tested. There's nothing to say that plague bacteria can remain active for even longer than 10 months maximum, let alone 7 years. But it causes all sorts of distress and problems in controlling outbreaks as well, because people want to avoid going to the hospital. Will be, where the state are going to take control. So this whole idea of the plague underground has been extremely important politically and remains important because they've never changed that policy. Until now, finally, the Pasteur Institute is doing a rat experiment where they're injecting plague into rats, burying them in barrels full of earth, and then they're exhuming them after a month, after 2 months, to see if there's any live plague bacteria. And finally, they'll have the data. And on the basis of this data, they'll be able to, I hope, revise the mortuary policy, because that causes a lot of heartache. But this is all of this interest in rat burrows and whether rats tunnel towards corpses underground, or whether, this isn't just intellectually interesting to me, but it's very much about people's distress and the levels of fear of even going to the hospital, because if you can't be buried in the tomb, then you don't become an ancestor. If you don't become an ancestor, then you remain a ghost that haunts the living. So it's all very present and real to people today. It's not just scientific questions that are academic at this point. They're really important to people. So.
Thanks, Genese. So 2 hands are up. I'll let Ben go first since he hasn't asked a question yet.
Oh, I just wanted to add a few points to what you said. I thought it was really fascinating to think about the burial policy and the latency or dormancy of Yersinia pestis underground. What I've heard in Tanzania is that the reason why population ecology of plague doesn't always work is because plague, the bacteria, can actually lay dormant for an unknown number of years. So even if you have all these Eltonian population ecological predictions of the ups and downs of population, whether of the fleas or the rats, you still cannot know when the next plague is going to break out because of this dormancy effect. So I just thought I'd share that I've heard when talking to people. And then this other thing, my research wasn't really focused on plagues. I didn't really take good notes when I was in the archives, but I remember coming across a folder about Madagascar and how and this was during the 20s and the 30s of the 20th century, and how Madagascar was a port station for ships to dock while they get fumigated so that they would kill all the rats. And this was an attempt to stop the spread of plague from different ports at the time, like from Hong Kong and from Mumbai. So I thought that was also interesting, maybe for your work, if you're looking that up. But I didn't take notes, so I don't know which folder be in because I wasn't focused on plague at the time.
Yeah, that's really interesting. I haven't come across the port of Tamatave being a fumigation port. But I did hear a paper about that happening in the turn of the 20th century. And in fact, so Madagascar's port was another one hit during the sort of late 19th century network of ships and colonial ports of call. But there was an interesting paper too, because of course rats swim very well and they could easily, jump off a ship and avoid as soon as they get to the port. But I haven't seen that about Madagascar in particular, so that's really interesting too. But by that time, the towns were, after the first infection in 1898, there were already rats that had brought the plague to the island and were already in, in towns and villages. So I don't know if they could have controlled it from ships much after that. What was
Ben,
something?
go ahead.
I just want to say this idea about plague being dormant is really fascinating to me, because I haven't seen any evidence of that. But there's really contradictory mortuary policies now. As I said, when someone dies of the plague, they may not be buried in the sort of earthen subterranean tomb with a masonry, upper level, but they have to be buried in a separate, really it's a pit, like just a grave, often in a potter's field, often with multiple bodies, multiple victims thrown in there. And the idea is that if these buried bodies are away from the tomb, then people will not practice this ritual of secondary burial where they exhume the remains after the corpses have dried out, and they do another ceremony, because there was this fear that the secondary burial rituals could reactivate a plague. But the weird thing is there's also this idea that hypothesis that rats tunnel towards cadavers. So if you're burying people in just pits in the ground without coffins or anything, and if you also believe that human remains can contain live plague bacteria, then essentially you are allowing rats to tunnel towards the corpses and potentially absorb more plague bacteria and then, infect other subpopulations of rats. So I don't think their solution these many decades, it doesn't seem to correspond to their hypotheses about how the plague remains persistent underground. So there's all sorts of strange and contradictory policies around burial that they're still contending with. But the science hasn't been, there's still open questions. So about this dormancy, I haven't really seen evidence of that in any articles. So I'd be curious. And I think that was always just a hypothesis from the colonial period, but without any firm answers up until now, at least what I've read so far. If you have other sources, I'd love to see that. But this is something they're trying to answer still, which is, I hope they do.
Super
I'll just
interesting.
quickly say I've only heard about the dormancy thing from scientists working in Tanzania currently, and I'll send you an email, and then maybe if it's
that'll be great. Thank you.
But I haven't found it published anywhere either. So.
Between what they're doing in this scientists in the plague unit of the Pasteur Institute in Madagascar, maybe they'll have really fascinating new information. Thank you. It's still very cutting edge. It's really strange, but yeah.
I'll let Ben have the last question here.
Okay, thank you. And I'm no kind of rat expert, so I just have a thought really on there's another question here of, I guess, the kind of play on the seen and the unseen. And across many of the examples that you've given, there's kind of, I guess, a problem of representation, or particularly how visual representation operates. So you had kind of the slides in the laboratory. You had the question of whether or not you could take photographs. I was wondering what pressures are kind of operate. Oh, also the architectural question is sort of a question of what is seen and what is not seen. I was just wondering whether you had any thoughts on I guess the limits of seeing rats, or the technologies through which the relationship between rats and fleas and humans is made visible or visualized.
That's an interesting question. At times rats are. It has to do really with this commensal relationship. The more habituated rats are in certain areas to humans, they can be very bold in broad daylight and have no fear and really be a nuisance to people. But for the most part, what you see about rats in people's homes and in their homesteads is strategies to trap or keep rats away, and the architectures of their silos and in their homes, and in the way that they hang their belongings on the beams of their homes in baskets, and the way that rat tunnels perforate the mud and dung dwellings that they have. You know, rats are not visible when you can see in broad daylight generally, but people live strategically with rats to protect their belongings and their rice, unless rats are sick. And that was that one article I published. When there's an epizootic and rats are sick, that's when they behave bizarrely and come out in daylight and have these erratic mannerisms. But people have been used to seeing rats that are poisoned, which also act in the same way, so that they don't read it as plague, they've read it as poisoned rats. But at night is when people hear rats. You know, it's when you do hear rats, and it's when you hear their little feet scampering or them gnawing on things. So it's this world that comes alive at night, and that become. It's interesting to think like that. The relationship between humans and rats is sort of very animated at night, but also invisible, just because it's dark. Yeah, and I'm also interested in. There's a French ethnographer who made very beautiful illustrations of the very this collection of different types of rat traps that people around the island make in different styles. And a family that I visited who had lost several relatives to a plague outbreak had the father had fashioned this little rat trap at this hole in his house, partly as a game. But I mean, those kinds of things I find really fascinating, how people are living with rats, but in an antagonistic way, but still ingenious way, you know, both of them trying to outwit one another. And again, that's, you know, that is somewhat more visible than the flea situation, where you only feel after the fact of being bitten. But so that's a greater challenge as a human to see or to try to investigate that. So I don't know, I'm just relying on the science for that, I suppose.
So thank you so much, Genese. This has been a delight to discuss this paper. You know, it's hard to say delight when you're talking about, you know, fleas that are frantically searching for blood and plague and death, but it really was a smart paper. Next week, in the evening slot for Melbourne, early slot for Europe, Warwick Anderson, who is here today, will be talking about militarism and COVID, but in a very concrete way. So he got a call from a general who asked him how to handle the coronavirus pandemic. And so Warwick will be discussing how he found himself in this strange position of giving advice to the military about wow. COVID. So thanks again, Genese. Hope you come back to the reading group. Again, a real delight to engage with your paper.
I thank you all for such brilliant and really helpful comments and references. I really appreciate it, because this is very much still in development and I'm learning still, but it's been such a pleasure, and I would love to attend the next talk. Thank you so much, Evan and Rachel, for the invitation. Bye, everybody.
Thank you. Thanks. Bye, everyone.
Bye. See you next